Good day, Mr. Stoller here. Today we are going to look into DNA code of life, the great war of DNA code of life, and we're going to look at two different nucleic acids. And this nucleic acid includes your DNA, and your DNA is deoxyribonucleic acid, okay? And your RNA is your ribonucleic acid. Okay, so we're going to look into this, these two nucleic acids. Let's see here with the introduction here, what we need to understand here. So the brief history of the discovery of DNA. So the first two guys to know the structure of the DNA was the Franklin and Wilkins. But who discovered the 3D model that we now have is Watson and Crick. Watson and Crick discovered the 3D model, a double helix shape model that we are dealing with in today, what you call studies. Okay, so this is what we have. And now let's look at different types of cells that we have. Is your animal cell and your plant cell. So on these two cells, I just need to look into a few organisms that are important that we're going to be dealing with. So we've got these dots that you see here, these small dots that you see here, that's your ribosomes. That's your ribosomes. And what do your ribosomes do? This is the site for protein synthesis. So site for protein synthesis. Okay, then we have the mitochondria. We have the mitochondria here, and you can also see the mitochondria there. So we have the mitochondria there. Then let me just put it down this side. Okay, so we have the mitochondria. And your mitochondria, that's your source of your energy of a cell, source of energy. Then once you see mitochondria, you must think of it as a source of energy. Then in today's lecture, we need to know that is a site for your DNA. So some of your DNA, you find it in the in the mitochondria. So we need those that I'm putting ticks where you find your DNA. Then we have got your what you call chloroplast. Only in plants, chloroplast is only in plant. Also, this is your site for your DNA. Okay, then most of the DNA is found in the nucleus. So this is your nucleus, this is your nucleus. So your what is it? Let me just put it nucleus. So where I'm putting ticks there, you must remember that those are your site for your DNA, location of DNA, where DNA is located. Okay, so let's move on and see and understand what your DNA is. Now we are saying the DNA constitutes what we call the hereditary information. It contains hereditary information, and your DNA is made up of genes, different genes that are linked together, and these genes code or control specific characteristics in our body. So we are saying segments of what you call DNA. A segment of DNA is what you call a gene. A segment of DNA is what you call a gene. And it controls specific characteristics and also controls what you call the synthesis of specific proteins. So we'll understand that when we are doing protein synthesis. Now, so we are saying in the nucleus, almost every cell in your body is a collection of DNA needed to make you. So DNA make up where we are. Now DNA is in the nucleus is grouped into 23 sets of chromosomes that are called genome. So we understand that humans have got 46 chromosomes, and that's what we call a diploid set. So in this case, it's 46 chromosomes. 46 chromosomes, and these sets are your pairs of chromosomes. So it's 23 pairs of chromosomes, and the instruction to make your body a whole body and keep it working is contained in the DNA. That's what we need to understand. And now, if we check ourselves, when we look at ourselves, we see that we look similar to our uncles, your grandmothers, your parents as well, your brothers and sisters. Is because of the DNA, because the information that is carried in the DNA. So it's inherited to the next generation. Okay. So in your family line, you have certain characteristics that are passed on from generation to generation. What keeps it constant? What makes it consistent like that is because of the DNA. What we call preserving that information. Now we are saying instructions is called the genetic code, and the DNA in your gene tells the cell what amino acid or protein building blocks to put together to make a protein. So we are saying your instruction to make proteins is actually comes from the gene, from the DNA, from the DNA. And what is it? Oh, just another thing here that I've not mentioned. RNA is single-stranded. We're going to look at it as well later in this lecture. Then also this one is what you call double-stranded. Your DNA is double-stranded. Now, what are the functions of the DNA that we need to know? DNA makes up genes which carry hereditary information. So like what I've explained, information that is passed on from generation to generation. Then also DNA contains coded information for protein synthesis. So it's very much important what call when it comes to synthesis of proteins. That's where the information or the instructions to produce proteins actually comes from. Okay, so we can actually say your DNA controls cell functioning. Then let's move on to our next slide here and see what we have. Now we need to know the structure of the DNA. Now we can see that it's coiled. So the coiling here is what we call the double helix. So we say the DNA is double helix. Let me just use a different color there. DNA is double helix. Okay, meaning to say it's coiled. Then what we also see is that it's double-stranded. So it constitutes of two strands linked together by weak hydrogen bonds. So it's linked together by weak hydrogen bonds. So that's what we see here. We can see that these are what we call two strands that are linked together. These are our two strands that are linked together. But also let's understand, we can see that we have A, T, G, C. What are these things that we're seeing here? Okay, let's understand something here. DNA is a polymer. So DNA is a polymer. When you're saying it's a polymer, it's a long chain, okay, made up of monomers. And the monomers are your building blocks. Building blocks of DNA. And these building blocks are called nucleotides. Now we need to understand that each nucleotide, how does it look like? How does each nucleotide looks like? We see that we have what we call a phosphate group. Then we have the deoxyribose sugar here. Then we have a nitrogenous base there. Okay, so we have got the phosphate group. Phosphate. group. Then we have got the deoxyribose sugar. Deoxyribose sugar. Then we also have what we call nitrogenous. Now, each nucleotide contains one nitrogenous base, one phosphate group, and also one, deoxyribose sugar. Now, there are four choices here. There are four choices here that we can have under nitrogenous bases. We can have guanine, we can have cytosine, we can have adenine, we can have thymine. And these we can represent it with letter G, and here letter C, then here letter A, and here letter T. That's what we see also in the structure there, that we have got ATGC. But now, what do we need? What is this ACGC? Okay, we'll answer that. Let's also understand amongst these bases, we have got what we call the purine bases. And these are large molecules, and that include your adenine and guanine. Then we have got the pyrimidines, bases. These are small. And what are these? It's your cytosine and thymine. And these bases, they pair through what we call complementary base pairing or specific base pairing. So what we see here, we see what we call specific base pairing. Specific base pairing. Meaning to say your A can only pairs with the T, and your G can only pairs with the C. So adenine can only pair with thymine, and guanine can only pairs with what we call cytosine. That's what we have. And what we can also see is that with A and T, we see two hydrogen bonds, weak hydrogen bonds, and between G and C, we see three weak hydrogen bonds. That's also what we can see there. We can also tell that, oh, what we have here is guanine and cytosine just by looking at the hydrogen bonds that we're having, and we can also see that there are two there. Now, what else do we need? To understand there? Now we know the specific base pairing. Now we know we've got the big bases and small bases. But also another thing is that these adenine and thymine, they're always in equal numbers. So we have got equal bases, equal amount of bases that we have, equal amount of bases, which is your A is equal to your T, then also your G is equal to your C. So since they are pairing, number of adenine are always equal to thymine, number of guanine are always equal to cytosine, because these two has to pair. So we can't have only one, like an extra thymine without a pair. We always have same numbers for complementary bases. Okay, that's what we need to know about the structure of the DNA. Let's move on to the next slide. Then we need to know that DNA replicate, making copies of itself. When I think DNA replication, when DNA make copies of itself. Now, the question is, when does this happen? When does this happen? That's the first question here that we have. So DNA replication occurs usually before, cell division, before cell division. So in the cell cycle, in cell cycle, it occurs during interphase, in the cell cycle. In the cell cycle. Okay, so it occurs during what you call interphase, during cell cycle, and this is part of what you call preparation for cell division, because we need to have enough DNA before cell can divide, because once cell divide, they need to share the DNA. And how are they going to share the DNA if they don't have enough DNA? It's more like we want to distribute copies of textbooks at a school. If we don't have enough, we need to make copies. We need to get the copies of of the same book. And now that's what you're actually doing here, to say during interphase, the cell is preparing for cell division. The cell is preparing, it's part of growing. When it's fully grown, it now has to make enough DNA, so that when it makes its copies of DNA, is the DNA can be splitted into daughter cells. Now, where do this actually happen? So we know that DNA is where it's actually in our nucleus, and that's where your DNA replication occurs. So DNA replication occurs in the nucleus. That's where DNA replication make copies of itself, in the nucleus. Now, how does this process go about? Now, we need to understand something here, that DNA is double helix. Now, if it's double helix, it means that it's coiled. So what needs to happen first is it has to unwind. So DNA unwind. Okay, so you can see that it's actually coiled. It's actually coiled, and it has to unwind first. Then the second thing that needs to happen, you can see that your bases here, the bases are linked together by weak hydrogen bonds. Now it's straight. It's now straight. It unwinded, but now we still have two strands attached to each other. So weak hydrogen. Sorry about that. Weak hydrogen bond breaks. Now, when the weak hydrogen bond breaks, what happens? Your DNA unzip. So it now unzips into two separate strands. You can see that you have got one here. Let me just use maybe a red color there. We have got one there, and we also have another strand here, which is two. These are original strands. So DNA unzip. What is this spelling now that I'm putting here? Let me just correct here. Unzip. Okay. DNA unzip. It separate. Now separate into two strands now. Now when you separate into two strands, we are saying each strand act as a template, because we are going to produce our new copies there from from those templates. So we are going to copy the information from the original template. That's what we're going to use. We're going to use to build a new strand. We're going to use the old strands, and that's what we call a template. So each strand acts as a template. Now, we have got free nucleotides here. You can see that we've got free nucleotides here that are in the nucleoplasm. And these free nucleotides attaches to what call to their complementary bases. So attaches to a basis of original strand, and through what we call specific base pairing. So free nucleotides build the complementary strand, which is this, it's a new strand. So build the complementary strand through, but it's explaining specific base pairing. And what is this specific base pairing? A only pairs with T. Let me just use a different color there. A only pairs with T, and your, what is it? And G only pairs with C. That's what we see. And now we are saying after that, what now do we have? We have got two identical strands now, because we are saying after this, we will have two identical strands. So two identical strands are formed. Identical DNA molecules, sorry. Molecules are formed, each constituting of one old strand, one old and one new strand. So we'll have an old strand. So what we can see here, we are seeing that this is a new strand. Let me do this here. Okay, so you can see that this is a new strand. And this one is an old one. So this one is an old strand that we can see here, and this one is a new strand that we are forming here. And in the end, we have got two identical DNA molecules. And now that's what we call DNA replication. So in this case now, we have two identical molecules, and the process is done. Now, why do we have DNA replication? Why do we have DNA replication? Number 4. We need to understand that DNA replication is important, what you call to make enough DNA for the cells. So here we are saying so we are saying this doubles genetic material so it can be shared. Daughter cells. Of daughter cells during cell division, but now daughter cells, during cell division, daughter cells during cell division. And again, what we need to also understand is that it results in the formation of identical daughter cells during mitosis. So formation of daughter cells, formation of identical daughter cells during mitosis. Okay. And another thing that we also need to understand is that, remember, mutations occurs during mutations. So mutations occurs during what you call DNA replication, and this what you call brings variation. Okay, that's also another thing that we also need to understand. During process of what we call DNA replication, some mistakes happens, like in the what call in the copying of copying of your DNA. And now what happens now is that it will bring variation. That's why we look different. We look slightly different. We have certain what call characteristics that we carry on ourselves that actually identifies us. And that's what we call mutations. And some mutations are beneficial, some mutations are also harmful. But now let's look at what we call DNA profiling. Now, DNA profiling is a technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. I think you now what is it where what you call you've read about things like people saying his DNA was found, or DNA test is testing for paternity test. Why don't you go for DNA test if you don't trust? If you're looking for your father or you're looking for your family, you can do what you call DNA test. And those DNA tests, it actually shows you who are you related to, and also it can also identify you as well as an individual during crimes. So if you found to commit crime, police, what they do, they take fingerprints, and when they take fingerprints, they compare it on the suspect list with the suspect list. So if one of them matches with the DNA on the scene, it means that what you call that's the suspect, and that's actually the person that they want. So in this case here, we're going to talk about the uses of DNA profilings. And use of DNA is for crime, solving crimes. And also another thing is paternity test. So paternity, you want to know who is your father. Like for example, here we've got the mother and the child, and you've got dead 1, dead 2, dead 3. We know that the mother always knows that that's my child, but now the question comes on the fathers, because you might see that what you call the parent is not sure who the child what you call belongs to. So in this case here, we can see we have got the child here, and we have got its own bands here, and we see that we have got number 1 daddy, 2, and number 3. Which one do you think is the father here? Which one do you think is the father here? I want you guys to comment and tell me which one do you think is the father what you call of this. Now, another thing also that you can also use to DNA profiling is finding relatives. And lastly, there is what you call possibilities of genetic disorders. So you can also check if if you have high chance of maybe carrying certain recessive what you call harmful alleles, then that's actually what you're talking about here, possibility of genetic what you call disorders. That's what we some people is, but it's expensive. DNA profiling is expensive, and some people do want to know what they could have in their gene. And normally couples who are rich, they can also go and try to find out if there are possible what you call disorders of their children. Now let's look at the RNA, which is ribonucleic acid, and we need to know the location of RNA. So we can call this one here 1. We call this 1, 2, 3. So let's look at number 1 there. Location, where do we find our RNA? RNA, we've got different types of RNA. We have what called messenger RNA, and we have what called transfer RNA and ribosomal RNA. So now when you look at messenger RNA carries the code of what you call for protein synthesis from your nucleus to your ribosomes. Now, your messenger RNA is found in the nucleus, it's formed in the nucleus. So it's formed in nucleus. Then and also move. Sorry about that. Nucleus and move to the cytoplasm, to the ribosomes. Where are the ribosomes? In the cytoplasm. Now, what about transfer RNA? Transfer RNA carries the amino acids to the ribosomes. So where do we find them? We find them in the cytoplasm. Okay, then we have got what called ribosomal RNA, rRNA. That's your on the what is it on the ribosomes, and this one is where we are saying site for photosynthesis, site for what is it, for protein synthesis. Now, what is the structure of the DNA? So let's look at number 2 there. Structure of the DNA is single-stranded, okay, and single-stranded it's made up of also, it's a polymer made up of monomers, nucleotides, okay? Then the other thing that we need to know is that each nucleotide, in this case now, we have got the phosphate group, we have the, in this case, we are no longer have the deoxyribose sugar, we have the, what is it, what we call ribose sugar. So here we have the phosphate group, then we have got the ribose sugar, then you have the nitrogenous base. Now, under nitrogenous bases, we have got 4 different types of nitrogenous bases. We have got the, what is it, uracil, we have adenine, we have guanine, and we have cytosine. Now, these are represented as U, A, G, and C. Okay. Now, in this case now, we need to understand that we have replaced the thymine with uracil. So in the RNA, there is no thymine, there is what we call the uracil, and what is it? That's all. That's what you need to understand. Then let's look at number 3. Number 3, we need to know the functioning. We know that messenger RNA carries the code for synthesis of proteins from the nucleus to the ribosomes. Okay, then you have the transfer RNA carries nucleic acid, amino acid, carries amino acids. Now, amino acids are building blocks of proteins. They carries amino acids to the ribosomes. Which is the site for protein synthesis. Then you have the rRNA, which actually assists in the protein synthesis. So here is your site for protein synthesis. That's what you need to know about RNA. Okay. Let's move on to the last part, last slide, where we compare the DNA and the RNA. What we see is that your DNA, we have the DNA here, then we have the RNA there. And what we see is that your DNA is double helix, is double stranded. So double stranded, double helix, and also double stranded. This RNA is single stranded. Okay. Then we have got thymine present here. We have got thymine base. Here we have got uracil present there. Then also another thing that we need to know is that we have deoxyribose sugar. And here we have got the ribose sugar. Then here it can replicate itself. Can replicate itself. Cannot do that. So we are saying DNA can make itself, can replicate, can make copies of itself, cannot replicate. Okay, that's what we need to understand. So we can see that we have the thymine here. I'm just gonna put mark here. Then here we have got, what is it? The uracil. That's what we need to see here. And you can see that the RNA is single-stranded, DNA is double-stranded. When you see double-stranded, a double strand like this, you know that DNA. When you see single-strand, you know that what you call is RNA. Then also whenever you see uracil, you know that you're dealing with RNA. When you see thymine, you know that you're dealing with what you call DNA. So here is something that you need to note, and also the double strand here and the single strand there. And on the deoxyribose sugar, unless if it's stated, you can't see that. If the nucleotide is a ribose, the sugar is deoxyribose or ribose sugar. But what you can quickly see is the thymine, presence of thymine or presence of uracil, double strand or a single strand. Okay. Thank you so much, guys. Until next time, all the best.